So recording has started, and I hope my screen is visible to you all.
Yes, ma'am.
Okay, so the things that I'll talk about today, you can easily find in any cell culture book. Okay, like in more details. I have even mentioned the book name that you can go through later on. But like since we have less amount of time, so we will cover only few things, and that too as in and as brief as possible. Okay. So when you are talking about cell or tissue culture, if you don't do it, you won't be able to make the tissue engineered medical products. It is the cornerstone for making these products. So the techniques that are used for fundamental investigation uh, for for the determination of the cell fate and how the cell or the tissue is going to mature in vitro. Okay, then how are you going to maintain the tissue construct and transport it? So when you're trying to understand all these questions related to a medical product, you have to look back to what are the techniques that are used for culturing cells. The similar techniques can be used when you're trying to maintain tissue engineered medical products. So there are various advantages of tissue cultures. First is you can accurately control the physiological environment, like what will be the pH, CO2, O2 level. Okay. Also, the sample, that is the cells that are using, is homogeneous. You are culturing only one type of cell at a time, so homogeneity is there. And as well as the cells that are using are well characterized. So they use various kind of characterization techniques like flow cytometer, etc., to see whether the specific cell surface markers are being recognized, that is used to identify that particular cell. And also, in relatively, if you culture cells, it is cheaper. Okay, rather than if you use models like animal models, it's going to be more expensive for like screening. Like if you want to screen any drug, the first thing that you should do is test your drug activity in vitro, like using cells, and then a more expensive way of proving the same thing is going via in vivo techniques. But it has few disadvantages as well. Like the sample size is very small, like where you're dealing with very small number of cells. Scale up can be challenging. Then the primary cells, they can exhibit functions that are not present in vivo. So you can, and also their functions or morphology, it can vary over length of time over which they are cultivated. So the cell behavior can change over time. Okay, when you are culturing it in using tissue culture techniques. So there are few types of cultures, tissue cultures. First is the primary cultures. The primary cultures refers to those cells that are directly isolated from the body, either animal body or human body, like directly from animal tissue or taken out the cells. That is primary culture. And then you seed it on a substrate. If it is adherent cell, it is going to need a substrate on which it is going to adhere, and then they will divide. And after few number of divisions, cell divisions, you will have to transfer the cells to a new vessel. Okay, that is to a new substrate. That is called passing. So you are passing from one vessel, few cells, to another vessel. Okay, when so some cells they grow very well even after large number of passings. So when they are able to do it, they become a cell line. Okay, so if the cell was initially taken from tissue, animal tissue, then it is going to be called a primary cell line. But the limitation is with this primary cell line is they will only grow for a limited number of generations. And if you want to add some modifications to these cells, like through virus or etc., some transformations, then the cell will become immortal, means it will grow continuously over a long period of time, over a large number of passings. Okay, so that is immortalized cell line. So when you are maintaining cells and you see the easier, which type of cell is easier to maintain, then it is the immortalized cell lines that are easier to maintain than the primary cells or primary cell lines, because these primary cells, as I said, they show some changes over a large number of passings, like the changes in gene expression or cell morphology. Okay, so in because of which when you are using these cells for an experiment, okay, then you may not see reproducible results. So cells from a one passage number, the result is different. When you're using the same cell but from a higher passing number, then the effect, the result is different. But when you compare this with the immortalized cell line, which grows continuously, then the reproducibility in your results become higher. So if you guys have any queries, okay, you guys let me know, because I'll go a bit fast because I have to cover a lot of topic in today's class. So in between, if you guys have any queries, please feel free to stop me and ask. So some primary cells, they can be transformed into immortalized cell line, as I said, by uptake of new genetic material. Means you introduce a new gene, okay, that can get embedded into its chromosome and generate new set of proteins that will give the ability to grow continuously, to become immortal. But when you are doing immortalization, okay, then genetic instability results. Means the some mutation will arise, okay, and some important proteins like RB, P53, these proteins, they will not function. So the genome or the genotype of the cell is not stable. It is rather exposed to changes. It can change very easily. That is called genetic instability. So when you are introducing this kind of genetic instability to a primary cell and make it immortalized, the some problems that can happen is it can become malignant. Okay, so these cells become cancerous cells. So you can these cells to make tumor models and do experiments like screen a drug for anti-cancer effects. And also these cells do not have the contact inhibition in them, so they grow continuously. Even when the surface is like complete, they can grow one over the other in a multi-layer fashion. Okay, now there are a few types of primary culture. So what I said, what is primary culture? Primary culture is when you're taking cells directly from an animal tissue. So there are three main types of doing primary culture. So you can do it from an organ. So in this figure, so you can directly do it like from an organ, okay, or you can, it is called organ culture, or you can do primary explant culture, and third is cell culture. So main difference between these three types of culture is, suppose you have taken out a small section of a tissue from an animal body and allowing that tissue to adhere on a cellular friendly substrate on which the cell growth is supported. You are allowing that whole tissue to adhere on that substrate, and over the time this tissue starts flourishing. Okay, it receives nutrients and shows the desired effects that you want. Wait a second. Sorry for the interruption. So as I was saying, you basically take a small tissue section from the animal body and place it in a suitable 3D environment where the conditions are optimized, in which you are basically growing that tissue. Okay, so that is called organ culture. In primary explant culture, what you do, you take that tissue, rather than growing it as a whole, you fragment it. Okay, cut it, chop it up into small pieces, and then allow those small pieces to grow in a suitable condition. Okay, and last type of culture, what you do, you chop up the tissue. Okay, you chop up the tissue into few fragments, or you take the whole tissue and enzymatically degrade whatever ECM component is there in order to free up the cells individually. Okay, the cells will get freed up and they become individual cells, and you get a cell suspension where the cells are floating in a liquid, and then use this cell suspension to like you seed it on a suitable substrate on which these cells will now start growing. So these three types of primary cultures are possible. But when we are talking about tissue culture, we refer to both the 3D organ culture, that where we are taking the whole tissue, as well as the cell culture where we are taking individual cells as starter for uh, growing, for the growth. So here is an example. You like take a tissue, okay, you from an organism, like from an embryo or from an adult organism or from egg, okay, that is zygote, fertilized zygote. So you take that and then you dissect it. So you can either enzymatically degrade it to get individual cells and do cell culture, or you like take that entire tissue construct and grow it on a particular substrate, that is organ culture, or you finely chop it and then you place these chopped pieces on a particular substrate on which the cells that are there in the chopped pieces, they will start growing. So these are the three main types of primary culture. Now how do you make a cell line? So as I said, from primary culture you get cells, and these cells, they can be either primary cell lines or immortalized cell lines. Primary cell lines, they have limited they can be grown for a limited period of time, whereas immortalized cell lines, they can be grown for a long period of time. Okay, but how do you make these cell lines? So have a look at this figure. So suppose you have uh, at this time point at zeroth week, you have started your primary culture either from like from the tissue. I'm talking about the individual cells, means you are doing a cell culture, that and these cells are derived from a primary tissue. So you took that cell and did the primary culture at zeroth week, and you allow those cells to grow for say up to 2 weeks, and then you see that the cell growth is increasing in an exponential fashion, but it can only be grown up to a limited period of time, say 14th week, and during this 14th week you are changing the vessel. So the vessel that you are using is going to have a limited surface area. So when the surface area is completely occupied by the cells, you have to change the vessel. Okay, that is called subculturing or passing. Okay, or splitting, cell splitting is also called. So you are doing subculture 1, subculture 2, like this you are doing like 5, 8 to 10 times, and after 8 to 10 times of passing the cells or subculturing, you see that either the cell will start showing the signs of senescence, that is cell has become old, and more cells you will see are dead. Okay, it can either go to this route after say 14th week, or it can go to this route where the genotype of the cell, it has undergone genetic instability, like some kind of mutation has added up, because of which the cell line has spontaneously transformed. Okay, and due to this transformation, this cell has become a continuous cell line or an immortalized cell line. So up to here, when you are culturing the cell for say 8 to 10 passages, then this is your primary cell line having a finite lifetime. Whereas if the cell, if the genotype if in the genotype some mutation or some gene expression etc has changed, the cell has transformed and become a immortalized cell line. So let's read what is written here. So initially a primary culture is established from primary cell source. If one can successfully pass the primary culture, then a primary cell line is established, and such cell lines can only be passed for a limited number of times. And if the cell line is transformed successfully, like they have gone to this upward arrow route, then a continuous cell is established, that is a immortalized cell line. Now you can increase the speed at which an immortalized cell line will be established. How you will do it? The transformed cells can arise spontaneously by a few cells escaping senescence, while most cells in the culture die. So out of millions of cells, only one or two cells will get transformed, and rest all of the cells will die. Like that is the uh, like seen thing. Like that will happen naturally. But if you want that all million cells should go transformation, so that you can establish a flourishing immortalized cell line, then you can do it by introducing some chemical or some virus. Okay, so what happens during transformation? So it can means cells can transform due to loss of function of p53 protein or rb protein, or overexpression of the telomerase gene. So we will study the function of the telomerase enzyme when we'll talk about stem cells. Okay, and also there can be mutation in the genes that is responsible for causing senescence. So when we are introducing these viruses like SV40 large T antigen virus or whole, this is basically a small section of the viral genome, or you can also use the whole virus molecule. Okay, so you are using these virus genes or whole viruses or chemicals to induce the transformation process. So these virus genes, they will also do the same thing. They will either inhibit the activity of rb protein, 53 protein, and this kind of inhibition will increase the lifespan. It can also result in again changing the activity of telomerase enzyme etc. So why this rb protein or 53 protein is getting targeted here? For making the cell line immortalized. So if you remember, when we were talking about cell division, and we briefly touched upon cell cycle, you remember that the cell has to pass through some checkpoints, and these checkpoints are present in between each phase of the cell cycle. So these checkpoints analyze whether all the processes has completed in the cells or not before it is ready to move on to the next phase. So these checkpoint proteins like p53 rb, these are the checkpoint proteins, and they are very very important. So if suppose the cell is like escapes the checkpoint, it will like become cancerous. Okay, and it whatever uh, abnormality that was there, okay, uh, it will start multiplying. So the checkpoints ensure that these aberrant mutations in the cell doesn't progresses to further generations. They uh, initiates apoptosis in those cells immediately, in those mutated cells. Okay, and doesn't allow those mutated cells to further divide. So that is why these proteins are getting targeted when we are trying to immortalize the cell lines. So immortalized cell lines, as I said, they are easier to maintain in lab, but due to genetic instability, some key features in these type of cell line is aneuploidy. Means if you remember the Down syndrome, how the Down syndrome is, there is a trisomy of chromosome 21. So basically the chromosome 21, it has broken off. There are two chromosomes, right? chromosomes exist in pairs. So one of the chromosome 21 breaks off in between, making three chromosome 21. So in this way, there can be addition or subtraction of a chromosome, whole chromosome, and that kind of thing happens when you analyze the genome of immortalized cell lines. Also, various cell lines which are initially in the primary culture, they were adherent, like they needed a substrate for growth. Due to immortalization, they lose this ability to adhere, and as well as these cells gain contact in lose they lose contact inhibition, because of which they can form tumors. So these kind of features will come in when you are working with immortalized cell line. So the choice of the cell line basically depends on what kind of tissue you are making or what kind of experiments you are running. Okay, but you should keep in mind that these kind of things are happening inside the cell while you're working with it, because of which you can see some unexpected changes while you are working with these cells over a large period of time. Now, as I said, cells, the phenotype, the genotype of the cells, they can vary over the time. I've already like hinted on this. So more detail on this is given on this slide. So over time in culture, the cells can exhibit both genotypic and phenotypic drift. Most continuous cell lines, they contain a variety of genotypes. The genotypes can change either due to spontaneous chromosomal alterations like aneuploidy, right, where there's a gain or loss of chromosome, or due to transformation of previously stable genotype into the one that is unstable. Okay, to overcome this cell line instability or genetic instability, what one should do is use a standardized cell culture conditions. Okay, and this standardized conditions should be maintained throughout the uh, period of you are working with the cell. So if the cell is capable of going through 12 passages, so you have to maintain the same culture conditions for that cell, so that you do not cause genetic instability in your cell line. Also, you should always store stocks of your cells. So that is called cryopreservation, that I also talk about in today's class. So in case you see genetic instability happening, means there is a change in the phenotype of the cell, you can discard your passage that you're working with currently and use your cryopreserved cells to again initiate the cell line. So these kind of procedures, they needs to be enforced rigorously in order to achieve reproducibility in our experimental results, as well as for quality control purposes. So far I have discussed what are the types of cells that we use in tissue culture or cell culture. Okay, how do you establish a cell line, what are primary cell line, what are immortalized cell line. Okay, so how genetic instability happens in cell line, because of which immortalization of the cell lines can take place. Now we will talk about the media. Media is nothing but a liquid that is going to contain a lot of nutrients, that these which are important for supplying energy to these cells. Okay, and promote cell growth and cell function, and that too happening in vitro in a lab scale. That is your media or cell culture media. So any queries so far? I think these all things are pretty standard and theoretical basically, and that is why I'm going a bit fast. So if you guys have any queries, you should let me know by interrupting me at any time and ask your query. Okay. So these are in this table, these are the components that are typically found in a cell culture media. So first, like here, note this is a basal medium components, means in this medium there is like no serum. Okay, or that is like serum free medium, usually we call a basal medium. But basal means also the precursor media, you can also think of it like that. when we add some additional substituents like growth factors or antibiotics, etc., then the medium composition is complete. So in this way we distinguish between basal media and complete media. So what are the compounds that we give in basal media? Like salts, like sodium chloride and other inorganic salts, and sodium bicarbonate. So sodium bicarbonate is used for maintaining the pH of your medium, and the physiological pH is maintained, that is 7.4. So how this pH is maintained using sodium bicarbonate, I'll come to it little later. As well as there is glucose, various amino acids, vitamins, and phenol red. Okay, so these are the components of the basal media. Phenol red is basically a pH indicator, where at neutral pH of 7.4 the color is red, and as the medium becomes acidic, it goes transition like from orange to yellowish. Yellowish means it's completely acidic. So in this way you can visually understand how much what is the pH of your medium. And when you add additional factors like growth factors or hormones or serums or antibiotics, then your media becomes complete. So serum is usually used to promote cell growth, and also it contains various kind of proteins, okay, that stimulate cell growth. Then you can also add antibiotics. So it depends on the lab practices whether they are using antibiotics or not in the complete media. But for safe side, people do use antibiotics in order to prevent the growth of any microorganisms in your media. Because if your since your media has all the organic constituents like glucose, etc., it can also attract the growth of microorganisms as well, like bacteria, yeast, etc. So if such kind of growth takes place, then there will be nutrient deficiency for your cell line, because of which their function and their everything will be compromised. Okay, you cannot work with those cells because the composition is no longer homogeneous. Your results won't be consistent if you are using a contaminated cell line. So you must take care that contamination doesn't happen. So we will also talk about contamination as well. Now, as I said, that you need sodium bicarbonate to regulate the pH of the media to 7.4. So how does it happen? So in order to control the pH, the cells, they are typically grown in a humidified incubator that maintains a gas phase CO2 of 5 to 10% together with sodium bicarbonate as medium additive. So you have added your cells in a vessel, and then you have to place it. So we place it in a CO2 incubator, humidified CO2 incubator. Means there should be some moisture inside the incubator, as well as continuous flow of CO2. And usually we maintain it at 5%, but people also keep it at 10%. And in the media, we add sodium bicarbonate. Why? So in water, so in the media there is obviously the main constituent water is present, and when this water comes in contact with the CO2, so CO2 in the like environment, it can easily get dissolved in the medium. So the dissolved CO2, when it comes in contact with water, it forms the carbonic acid. Okay, and this carbonic acid further releases the hydrogen ions, so because of which the pH is changing. If you remember how the pH is calculated, we use the concentration of hydrogen ions to calculate the pH. So more the hydrogen ion concentration, more the pH will be towards acidic. So this kind of reaction takes place. Now we want, in order to maintain the pH of 7.4, we want that always there should be high concentration of bicarbonate ion. Okay, means the reaction should not move towards the carbonic acid side, means towards the left side. If it happens, then the pH will become acidic. So in order to increase the concentration of the bicarbonate ion, we add sodium bicarbonate, which releases this excess bicarbonate ions. And if you want to calculate what is the pH of the given media and the incubator. So we are using an incubator having 5% CO2 with the media having, say, some grams of sodium bicarbonate that is going to release some amount of bicarbonate ions. So if you know these two values, then using that you can calculate the pH of your media using this equation. This equation is called Henderson-Hasselbalch equation. So basically this is, you don't need to do it every time. It's pretty standardized, and if you go to a lab, they will teach you like that you have to use this much of sodium bicarbonate for incubator that is set at, say, 5% or 10% CO2 level. So that is why we don't do this pH calculation every time. We know this is standardized, so we always follow it. So in our lab, what we do, if we are using the media DMEM, the full form of DMEM stands for Dalbacco's Modified Eagles Media. So this is one of the very common cell culture media that is used. So for this media, we whenever we use this media for culturing the cells, we use incubators with 5% CO2, and to this media we add 3.7 grams per liter of sodium bicarbonate. So when we do it, we achieve a pH level of 7.4 to 7.2. And at this pH level, the phenol red, that is the pH indicator that it is already there in the media, it turns red, okay, pinkish red. Now oxygen is also important, okay. So CO2 level, we are maintaining the pH of the media, okay, but oxygen is needed by the cells for its respiration, right? So oxygen delivery in culture, so we don't add additional oxygen cylinders, we don't hook up the incubator with oxygen cylinders because there is no need. In the air there is already oxygen, but these oxygen, it has to get dissolved in the media. So that is the main difference when you are culturing the cells in vitro in comparison to what is happening in vivo. So in vivo there is like RBCs that carry the oxygen molecules to the cells, but in vitro the oxygen has to get dissolved in the medium. So delivery of oxygen to the cells, they are limited by these two factors. First the solubility of oxygen in media, and second, how they are transported from the gas phase to culture media. So these two factors dictate how much oxygen is present to the cells that you are growing, okay. Now in general, oxygen doesn't have that much solubility in aqueous based solvents like media, which is why in our blood also we need carriers, hemoglobins, because oxygen solubility is very less. Similarly in media as well, solubility of oxygen is very low. So what would happen to the pH of the media if the cell line is grown at a high density? So under these circumstances, what will happen? Will the level of oxygen play any role? So if the cell density is very high, then the oxygen consumption per unit area of the cell will increase, okay, because of which there will be decrease in cell surface oxygen concentration. There will be less oxygen, the less dissolved oxygen available in the medium, and that will cause a shift from aerobic respiration to anaerobic respiration pathways. And you know, during anaerobic respiration in our body as well, it happens, there is a production of lactic acid, and this lactic acid production will cause a reduction in pH level, okay. So here excess production of CO2, if there's it's a cell at high cell density, there will be also high production of CO2, right? And as we have seen in this expression, if the CO2 level is very high, then it will move this reaction towards right hand side. That will also lead to the production of carbonic acid. So two types of acid will start getting produced, lactic acid and the carbonic acid. Together it will reduce the pH, and this reduction in pH you will able to see they will through the change in color of your pH indicator phenol red. It will become orangish to slightly yellow, okay, when the pH has become completely acidic. And at this acidic pH, the cells will not be able to grow. It will start coming out. It will start floating in the media, means the adherent capacity will lose slowly. This morphology of the cell will become abnormal, okay, and more you will see cells are dying. So whenever you see floating cells under microscope in the tissue culture, cell culture flask, it is, we usually associate that these cells are dead, okay. Dead cells can't adhere, as well as morphology starts changing. So always we try to ensure that the cell doesn't get in contact with the media which is highly acidic. We change the media with the fresh media in which the pH is 7.4. So this kind of standardization one needs to do in order to make your cells or maintain your cells in a healthy condition and reduce any variations that can happen over time. So any queries so far? I have only come halfway and I have halfway to go. So I apologize if today's class is going to go a bit like maybe I have to extend it a bit, but in today tomorrow there won't be a class, there will be only quiz, so maybe we can compensate like that. So now in the basal medium we add some serum. So what is a serum? First let me explain. The serum is basically a small fraction of the whole blood. So blood is composed of plasma and the cells, blood cells. So when you remove the cells, you get plasma. And the plasma, when you allow the plasma to coagulate, okay, you are left with serum. So coagulation will happen because of the formation of fibrin clots, right? So you have to centrifuge basically. A series of centrifugation steps will give you pure serum. So, and this serum we typically add around 1 to 20% by volume to our media. So in our lab we use 10% serum, and in our media 10% we use. And this the source of this serum or the sera can be multiple, like it can be from a calf, okay, bovine calf, a newborn bovine baby, or it can be a fetal bovine taken from the fetus, or it can be taken from a horse or from human. So which is why this serum is very expensive as well, okay. And they are taken directly from these animals, right? So we have to use it very consciously. This serum has many benefits. It contains important growth factors, adhesion factors, minerals, lipids, hormones, etc., which are very important for successful cell culture. But there can be disadvantages as well. So since you are using different animals, like to make say 100 of bottles, so composition of serum in 100 and in these hundreds of bottles will be slightly different from the next batch of 100 bottles that uh, it is sourced from another animal. So there is a batch to batch variation that can be present in serum. Also it can interfere, these variations can interfere with your experiments when you're trying to understand the production of any cell products, means secretion of any protein, etc., when you're trying to understand or to detect these kind of cell secreted products. So the serum proteins can go and interfere with your experiments, okay. So in general, as per my experience, this batch to batch variation doesn't have very significant role, okay. So far I haven't observed, but yes, batch to batch variation will be present from one batch of serum to another batch of serum. So these are the few few factors or the constituents that are present in the serum. Okay, I'll share with you the slides. You guys can go through them. Now you have to create an environment suitable for culturing your cells in vitro, that is outside the body. So how do you create this environment and how do you maintain this environment? The first and foremost thing, you need a sterile environment. Okay, cell culture cannot happen in open. Okay, you need a laminar hood where there is a laminar airflow going on. Okay, either in vertical direction or in horizontal direction. So this laminar airflow basically is a curtain of air, okay, that prevents entry of outside air inside your cabinet. So because of which you can create a sterile environment, and all the air that is flowing in the laminar airflow hood is passed through HEPA filters. So these filters are having a very small pore size, like 0.2 micron pore size, which is going to filter out all the airborne microbes. Okay, so the air that is circulating inside the hood, laminar airflow hood, is sterile, and the flow is of the air is, since is laminar, it is acting as a invisible curtain and preventing the entry of outside air. Okay, so laminar airflow hood is you needed. Also you need an incubator, and that incubator also need to be maintained in sterile conditions. You have to be very cautious while opening and closing it. Okay, you shouldn't open it for too long, otherwise outside airborne microbes can enter. Okay, so this kind of like things that you need to do in order to create a sterile environment. Also, entry should not be given to all personnels. People who are familiar with these protocols, only those people have to enter. So these sterile conditions you need to maintain. In many labs there are air showers, or there are special kind of dresses, okay, sterile dresses which are exposed to UV, okay, germicidal UV, and those you have to wear only those dress and pass through an air shower which will remove all the environmental dust particles, etc., so that the environment that you're going to enter, that is the cell culture facility, there no airborne dust or microbes, etc., is present. So these kind of sterile conditions you need to maintain first, then only you can proceed with culturing your cells. The cells can be of two types. They can either adhere, that is adherent cells, means their anchor is dependent cells, and other type of cell, that is the blood cells, etc., which do not need anchorage independent cells, okay, non-adherent cells. Now for culturing in any of these type of cell, you need to consider these parameters: soluble factors, ECM interactions, cell-cell interaction, physical factors. So these four factors we have already seen, right? How they are responsible for coordinating cellular fate processes. So similarly, since we are working with cells and we want all cellular fate processes to happen, like cell division, apoptosis, differentiation, etc., but in vitro condition, so you need to consider all these four parameters. Okay, and these four parameters I will now explain how they are maintained in vitro. And also the extra care is taken that we should mimic the in vivo condition, because the cells that you have taken is from a body, right, that was initially present. So from an in vivo condition, you are culturing these cells in vitro outside the body. So in order to get a stable functionality in your cell, you need to give it the similar conditions that was there in the body. Then only you will be able to do experiments, okay, and say that whatever drug or whatever tissue engineering medical product that are using is going to be suitable for in vivo application. So this logic, I think, is makes sense to you all, right? Now let's see all the parameters that I just said that needs to be defined for a tissue culture environment. The four parameters: the soluble factors, the ECM geometry and composition, cell-cell interaction, and the physicochemical factors. Soluble factors means the signaling molecules, the growth factors, etc. So they are brought into the culture through the media. So in the complete media, remember we have added the serum. The serum contains the growth factors basically. Okay, but if you need additional growth factors like special kind of growth factors, then you can also add in your complete media. And if you want a sustained release of growth factor, like gradually the level of growth factor will increase just like it happens in vivo. You have seen, right, how the growth factor concentration decreases from the signaling cell till it reaches the target cell through diffusion. So if you want to create or mimic that kind of condition, then you can use vehicles in which the growth factors are encapsulated, and through their pores, through the pores of these vehicles, there is a very slow release of growth factors. You can also do that. Then ECM. So ECM, cell ECM interaction, you will be able to control it by choosing what substrate you are using. So it is very important when you're talking about the adherent type of cells that need a substrate. So type of the substrate will dictate how the cell ECM interaction is going to take place in vitro. So people in general use either glass or plastic substrates. So it is safer to use the plastic one. And these plastic substrates, they are coated with polystyrene. Okay, first they are coated with a polymer called polystyrene, and this polystyrene is then modified. Okay, this layer of polystyrene is modified. How? It is exposed to ionized gases, okay, that add hydroxyl groups, which have, so due to the addition of hydroxyl groups, that is the OH group, the substrate becomes hydrophilic, okay, water loving substrate. And this kind of a hydrophilic substrate is essential for protein adhesion. Okay, so in the serum you have added, there are various kind of proteins, right, as I already said. So these proteins will adhere on a hydrophilic substrate, but not addition. Addition of the protein should happen in a proper configuration. Okay, it should not denature, it should be like the configuration that is normally present inside the body. So the configuration should be proper in original manner. So, and on top of that the cells will adhere. Okay, so that is the logic behind why we are making the substrate hydrophilic using a OH group modified polystyrene substrate. So we are trying to cause adsorption of the proteins in their original configuration that exposes the cell binding sites, and on top of which the cells will adhere, okay, and spread, and they will divide and differentiate, etc. Okay, now you may typically do it. So when you are using this kind of polystyrene, we also called it tissue culture polystyrene coated flask. Okay, so when you're using this tissue culture flask coated with polystyrene, you allow the adhesion of or adsorption of these serum proteins like vitronectin. Okay, but if you are using a serum free medium formulation, so in the complete media there is no serum, then what you do, you have to do a previous step, you have to do a prior step of coating this substrate with an ECM molecule like collagen or fibronectin. Okay, or you can coat it with peptides like RGD peptides, and then you seed the cells. Okay, because in the serum there is no protein, so there will be no addition of your proteins. So but you need proteins on which the cells will adhere. So in order to do that, you prior coat the flask substrate flask surface with a protein like collagen or fibronectin or a peptide. Okay. Now, when you add cell suspension, so these cells, they remain in round shape. Okay, and within few hours they adhere and take up their morphology. And if you see under microscope, they look like sunny side up, where you can see a dark colored nucleus. Okay, and few organelles like dot kind of a structure can be seen, but in electron microscope you can even see inside the organelles. That is not the thing that we do routinely, but routinely we observe, like once we add the cell, whether they are in round morphology, and after 4 or 5 hours when you again observe the cells have adhered and started taking their morphology. Now, cell cell interaction, they also influence how the cells are functioning in vitro. So if you are using only one type of cell, that is called homotypic culture, but if you are using multiple type of cell, then it is called heterotypic culture. Okay, so homotypic culture we generally do, and it is very relevant to human body as well. Like in epithelial tissues, there is a formation of tight junctions, and all the epithelial tissues are of same type. So this is a type of homotypic interaction that happens in vivo, and we are mimicking it in vitro when we are using similar type of epithelial cells. But heterotypic uh, interactions are also important, like how the function of fibroblasts are affected by the endothelial cells. Okay, or how the macrophages are influencing the functioning of, say, proliferating keratinocytes or fibroblasts, etc. So this kind of interactions, when you are going to study in vitro, then you need to do heterotypic interactions. So one such example where people use heterotypic interaction is, people generally use a sheet of fibroblast that is going to promote the growth of keratinocytes. So basically the fibroblasts and keratinocytes, they are grown at the same time. Okay, so any queries so far? No, no queries. Ketan, Shambhavi, are you guys there in the meeting?
Yes, ma'am.
So is there any query that on anything that has been
No,
discussed? No. Okay. Okay, so let's continue. So the soluble factors, cell ECM interaction, and cell cell interactions, they are all, you are going to maintain. These interactions are going to be preserved, or you are mimicking whatever happening in vivo in vitro conditions. But the physical parameters like the temperature and the physical forces, they also need to be taken care of when you are trying to create a tissue culture environment. So our body temperature is 37 degrees Celsius. So when you are culturing human cells, or say mouse cells, mouse body temperature is also close to 37 degrees Celsius. Then you need to maintain your incubators at 37 degrees Celsius. Okay. So, but if you want to metabolically reduce, means make the cells metabolically slow, then you can store the cells at 4 degrees Celsius for few days, but after some time, or after 3 4 days, the cells will start dying. But the optimum condition is to maintain the cells at 37 degrees Celsius, that is the physiological body temperature. But if you increase the temperature, say to 40 41 degrees Celsius, then what will happen? Then the cell will start expressing a special kind of protein called heat shock protein in order to protect itself. Okay, so you don't want this kind of stress response in your cells, which can affect your normal cell function. That is why we always maintain our incubator to 37 degrees Celsius. The physical forces like the fluid shear stresses, okay, then uniaxial or biaxial stretch. So if the substrate is getting stretched in uniaxial or biaxial directions, the normal pressures, so these all kind of forces also affect the cell functions. Like people have seen the flow in the direction, so when you are working with endothelial cells, so people have seen that the endothelial cells, they start growing or they start elongating in the direction in which it feels the fluid stress. Okay, so tangentially the fluid stress is there because of the flow of the fluid, so in that direction only the endothelial cells will spread, okay, and migrate. So that is like the effect that the physical forces like fluid shear stress has on cell function. And we have already seen that there is a class of biochemical signaling that is called mechanotransaction, where the physical forces like the shear stress, etc., get translated into biochemical reactions. And there are special type of receptors on the cells that can detect these mechanical signals. Okay. And so how fast the cells will grow, that is the kinetics. So if you I think you must have seen this type of a growth curve, cell growth curve, where initially there is a lag phase, then after a few days there will be a log phase where the cells is growing exponentially, and then there will be a plateau, okay, that is the stationary phase. So this kind of uh, growth curve can be also seen when you are culturing cells in vitro. Okay, so at say day 0 you have seeded some cell density, say you're starting with uh, 50,000 cells. Okay, so in for few hours you will see some lag phase, and then depending on what is the cell time, you will see the population doubling time. What is population doubling time? I'll come to it. So that population doubling time will vary depending on the cell type. But during this log phase, you'll be able to calculate what is the doubling time, population doubling time. And after each doubling time, the cell number will double basically. Okay, which is why you see an exponential rise in the cell number. Okay, and once the entire substrate is filled, okay, so you are growing the cells on a surface area say 25 centimeters square or 75 centimeters square. So there is a limited surface available for the cell growth. So when the cell number has reached to such level that entire substrate is occupied, then a plateau phase will take place, that is a stationary phase will occur. The similar kind of explanation can also be given when you are talking about cell suspensions, like the cells that grow in suspension form, the anchorage independent cells. You can also uh, give similar kind of explanation to understand how the growth curve is progressing. But in this case, the substrate is not limiting or is not causing the plateau phase. Rather, the media components, the secreted growth factors that are present in the media, so those are responsible for limiting or for deciding when the stationary phase will reach. So typically, cell culture media, it supports a growth of few million cells. Okay, but you can make special formulations that can support the growth of more than 10 million cells. So normally we use the first one, where growth of around 2 to 5 million cells is expected from a surface area of 75 centimeters square. Okay, so this slide which I have skipped is basically explaining the same thing that I have explained you using the graph. You guys can go through it once I share with you the slides. Now the next topic is cryopreservation. So what is cryopreservation? So you're using cryo, that is very at you're using very low temperatures, like negative temperatures, to preserve your cells. So it is necessary to keep the cells stored so that they can repeatedly be used from the same starting state. Now suppose you are culturing the cells for and you're using a primary cell culture. Okay, so using a primary cell having a finite life, and usually primary cells are very costly. One vial of cell having say 1 million cells usually cost around 50 to 1 lakh rupees. Okay, so it is not affordable to purchase every time once your cells have reached your finite stage, means after which the cells have started showing senescence. So it is not always possible to spend this much of money, and usually they reach senescence say within 1 month. So in that case, what we do, we make stocks, cell stocks, and these cell stocks are stored at very low temperatures like -80 degrees Celsius or -196 degrees Celsius, so that when we see that the cells have started undergoing senescence, we discard that passage number and we take out the cell stock from the storage, -80 degree storage or -196 storage, and restart the culturing from the same condition, like starting from passage number 1. Okay, so in this way we we use that cell line for years. That is the significance of cryopreservation. So -196 degrees Celsius is used for long term storage. That is the temperature of liquid nitrogen. Okay, that in liquid nitrogen we can store cells for 1 year, but not more than that. Also we can store it at -80 degrees Celsius. Now here the time for which we can store the cell is around 3 to 6 months. So every 3 to 6 months we need to again make new stocks, new cell stocks. So that is a routine thing that is done by the people who are working in cell culture. So some of the reasons, this is one reason that I told you why cryopreservation is needed. Other reasons is ability to recover a well characterized starting cell state for tissues and medical products, then prevent genotypic drift due to genetic instability, storage prior to senescence, prevention of transformation, control of phenotypic instability, a backup in case of contamination, and storage for characterization of pathogen testing. So these are some other reasons uh, that we consider for cryopreservation. Okay, so the first main few of the main reasons in this list is reason 1, 2, okay, 3 is also a good reason, 4, 5 and 6. So these 6 reasons, first 6 reasons are very important. The sixth reason, the backup, it is also a backup in case a microbial or a fungal or a virus contamination comes. Okay, so in that case we have to discard all the cells in our lab and again restart. So from where we will start? We cannot purchase the cells every time, especially when you are working in deadlines where your experiments are on hold. Okay, and you cannot devote your time waiting for cells to arrive. So in that cases, cryopreserved cell stocks are very important. Now the cells, they are removed from the substrate by enzymatic or mechanical disruption, and then you create a cell suspension. Okay, and this cell suspension is uh, dissolved in a solution containing a cryoprotectant. So what is cryoprotectant? Cryoprotectants are basically protecting the cells from very low temperature, and usually we use chemicals like DMSO, dimethyl sulfoxide, or grisolol as cryoprotectants. So these cryoprotectants, they prevent ice formation inside the cells, and if suppose ice formation takes place, then it can cause a large amount of cell death. So which is why cryoprotectants are very important to prevent cell death when cells are stored at a very low temperatures. Now, what is the effect of the cooling rate on cell survival during cryopreservation? So, you have to optimize what is the most efficient cooling rate, means how much at what is the temperature drop per minute. So, in our lab, we try to maintain a temperature drop of 1 degree Celsius per minute. So, in every minute, if we started with say 37 degrees Celsius, it will slowly drop to 80 degrees Celsius with 1 degree at a time. Okay, so this is called cooling rate, and this cooling rate has a effect on whether cells will have a survive, like whether cells will survive during this cryopreservation, means at a very low temperature. So cooling rate dictates the cell survival during cryopreservation. How? Okay, what is the logic behind it? So let's briefly explain that. So ice will start as soon as you place it inside say minus 80 degrees Celsius fridge or inside a liquid nitrogen tank. There the ice will start forming from the pure water, and the solutes, they will start accumulating in the unfrozen water, making the surrounding extracellular solution hypertonic. Okay, you understand hypertonic, right? Means solute concentration is high, so because of which the cells will undergo dehydration. So there will be osmosis, movement of water outside the cell. The cells will dehydrate. Due to this dehydration, there will be spontaneous nucleation of ice. Ice will start forming inside the dehydrated cells. Now initially, when the small ice crystals are formed, then the effect is not that much, means the cell damage is not that much. But the cells are extremely dehydrated, then there will be very high rate of cell death. There will be very fast cell damage. So even if you have stored your cells in liquid nitrogen thinking that your cells are safe and you can use it say after 1 year, but if the cooling rate is not optimum, then what will happen? The cell, because of this cell death that is happening due to the dehydration, when you revive your cell stock, none of the cells will adhere, all the cells will be dead. Okay, so this cooling rate has a very large effect on the cell survival during cryopreservation. So in this graph, if the if you are using optimum cooling rate, optimized cooling rate, then majority of the cells will survive. Okay, but due to solution effect, means due to dehydration of the cells, that will happen due to the creation of an hypertonic environment, you will see the cell survival percentage is declining. That is, on the left hand side of the graph is declining. So here the cooling rate is not optimum, because of that is it's happening. Another side is, if the cooling rate is very fast, so the first effect will happen, solution effect will happen if the cooling rate is very slow. And if you are using a very fast cooling rate, then what will happen? Then the intercellular water content, it cannot escape through osmosis, because of which large amount of ice will start forming inside the cell, and this iced means large amount of ice, they will start causing cell damage, basically cause cell perforations and basically make these cells dead, which is why you need a optimum cooling rate. And this cooling rate is optimum cooling rate exists for each type of cell, for each type of cryoprotectant solution. Okay, now once cells are frozen, that we have to thaw it. So when you are reviving, that is, we are thawing and reviving the stock. Suppose your lab is contaminated and you have thrown off all the cells, and again you have to start culturing the cells. So you have to take out the stocks from the liquid nitrogen tank and thaw it. So if you thaw it as soon as possible, then it is like expected that your cell survival rate will be high. Okay, because rapid thawing will minimize reformation of ice crystals inside the cell. So ice crystals, formation of ice crystals in the cell is the main what do you say, culprit that is causing cell death. So we have to always optimize the conditions so that the ice crystals doesn't form. Okay, so I was talking about this condition when the cooling rate is very fast, because of which ice starts forming inside the cell. So here also the cell survival rate is decreasing. The less and less number of cells will be alive. More and more cells will be dead in your stock. Now, the last topic in this discussion is contamination. Okay, so in the air there are in the air, in the water, like you are also a source of contamination, and contamination can uh, be like of many types. It can be either microbial, means it can be either bacterial, yeast, fungi, or mycoplasma and virus contamination. So these four types of contaminants are observed, or basically frequently observed when you're working with cells. So usually what happens, if contamination or if any bacteria started growing in your flask, then you will see a very rapid change in your pH. Means the pH will start becoming acidic. That is the first indicator. So if you have changed the media today and tomorrow you are going again, you have to change the media. Usually we change medias on alternate days, but you have to again, if you have to do it, then it is because some type of microbes, one of the reason could be some type of microbe has started growing cells. Okay, it will cause cell death, more and more cell death. So how will you detect the presence of these microbes? So bacteria can only be identified if you use a very powerful light microscope, where you can use magnification of 100 to 400 types. Means normal 10x or 40x is not sufficient. You have to go up to 100x to see bacteria growing. Whereas yeast, they can be easily observed as round particles. Okay, and they very rapidly increase over time. So these are the yeast. Whereas fungi, you can observe this fungal growth as filaments, these like small filaments growing, thin filaments growing in your media, or some spores that are growing. So the fungi and yeast, means basically the fungal infections, they are observable under microscope, whereas bacteria, it is very hard to detect. Whereas mycoplasma and virus, they are the most khatarnak type of contamination that one can imagine. It is a nightmare if a mycoplasma contamination comes into a lab, cell culture lab, so that the lab, because of which the lab can remain closed for a year, even a year, if the problem is not rectified immediately. Because these viruses or the mycoplasma, you cannot see it under naked eyes. They can also you know pass through the 0.2 micron filter papers. Okay, so you cannot even sterile filter your media or anything. So they are like very hard to catch, and they will gradually show its effect on the cells, like the morphology of the cells will start changing, or the growth rate will start changing. So you need special kind of assays in order to detect whether mycoplasma or viral infection is there or not. So what if suppose you find contamination in your cell line? Okay, so what steps you should do? First is immediately throw that cell line. If you detected that some unwanted host is there in your flask, tissue culture flask, then you should immediately throw it. Then you have to identify what could be the reason of arrival of this microbe in your flask? What could be the reason? You have to first identify that problem and then immediately rectify it if it is rectifiable. Okay, if it is if it is going to take some time, a safe side could be like do a chemical fumigation in your lab using chemicals like formaldehyde and potassium permanganate. So using these chemicals, you create an environment of toxic fumes. It is these fumes are so toxic that you have to close the seal of your lab. Okay, and should not do not should not inhale this toxic fume because this fume is also toxic for you, but it is 100% effective to kill off all the airborne microbes that might be there in your cell culture room. Okay, so and once your fumigation is done, okay, you will then again use you can then start reviving your cells from the liquid nitrogen tank and resume your cell culture work. But contamination is not desirable at all. You should always try to maintain sterile conditions so that this unwanted guest does not interfere your work, because then your work will be deeply hampered. So if you found this discussion interesting and want to learn more about it, so this book by Freshney, Culture of Animal Cells, a manual of basic technique. So this book is the Bible of cell culture, and in future if you ever going to do such kind of work, you can refer to this book in order to study about everything like media, cryopreservation, then how pH is maintained. Okay, everything you can find in more detail in this book. Okay, so in future you can definitely refer to this book if any of you are going for cell culture, in vitro cell culture type of work. So that's it. So I have taken 15 minutes extra today actually to finish up this topic, but I hope you have understood. And if there is any queries, you guys should let me know. Are there any queries? Any queries? Nidhi, Vani, are there any queries?
No, ma'am.
No. All right. So I'll share the slides with you guys. You should have like thorough reading of this slide. So and then you will understand better. And if you have any queries later as well, then you must let me know. So let me stop sharing and